हेलो फ्रेंड्स, मैं आप सभी का स्वागत करता हूं अपने ऑफिशियल यूट्यूब चैनल पे। एंड in today's lecture we are going to start about the nucleus architecture and organization. What is nucleus and what is its structure, as well as how the genetic material is oriented within the nucleus. किस प्रकार से न्यूक्लियस हमारी सेल के अंदर प्रेजेंट होता है और हमारे न्यूक्लियस के अंदर किस प्रकार से हमारा जेनेटिक मटेरियल क्रोमेटिन ऑर्गेनाइज होता है. सो प्लीज इफ वी कैन लुक फॉरवर्ड, द न्यूक्लियस वाज बीइंग डिस्कवर्ड बाय रॉबर्ट ब्राउन. J. Hammerling was the first person who firstly reported that nucleus acts as a controlling center of the cell. He was the first person who performed the experiment over the species of Estabularia crenulata and Estabularia mediterranea. He reversibly exchanged the nucleus in the foot state of the Estabularia crenulata into the mediterranea and from mediterranea into the Estabularia crenulata. You can see in the diagram there that it is very clear when the foot nucleus was being exchanged from Estabularia crenulata into Estabularia mediterranea. What we find is that the shape of the cap is actually being determined by the foot nucleus. Foot का जो nucleus है वही determine करता है shape of the cap को. Foot nucleus determines the shape of the cap. Experiment. The number of nucleus greatly varies. There are 2 in Paramecium chordatum and many in Paramecium multimicronucleatum. Cenocytic in the fungal hyphae of Rhizopus. What do you mean by cenocytic condition? Cenocytic is a type of condition when the karyokinesis is going on. Rapid nuclear division is taking place. Nucleus divide कर रहा है. Rapid nucleus division is taking place, but cytokinesis is not taking place. Then this condition is referred to as cenocytic condition. Another condition which may also you may find in books is referred to as syncytium condition of the nucleus, when the different cells are present. These cells are actually different in the origin, but later on the plasma membrane in the cell wall has dissolved. So the nucleus, it appears that there are many number of nucleus which are present. This condition is referred to as syncytium. Nucleus is present in all eukaryotic cells except the RBCs, sieve tube elements, xylem vesicles and tracheids. If nucleus is absent in any cell, then that cell is not going to survive for a longer period of time. तो वो सेल ज़्यादा समय तक के लिए सर्वाइव नहीं कर सकता. That is why the RBCs, the sieve tube elements, xylem vesicles, tracheids have a particular lifespan only. For example, RBCs have a normal lifespan of only 120 days. Now, the organization of the nucleus, how the nucleus is being organized, we can divide the nucleus into the following 4 parts. हम न्यूक्लियस को स्टडी करने के लिए following 4 parts में डिवाइड कर सकते हैं. जैसे कि हम सबसे पहला पार्ट जो पढ़ने वाले हैं, that is referred to as the nuclear envelope. Now, nuclear envelope consists of 2 layers. One is referred to as the exokaryotheca, the outermost layer. Another one is referred to as the endokaryotheca, the innermost layer. Okay, so as you can find, this board is there. This board has one layer on the surface. This would be referred to as endokaryotheca. This room, this interior area is referred to as this wall. Their walls is referred to as endokaryotheca. The exterior side will be referred to as the exokaryotheca. Now you can find there is a space in between the exokaryotheca and the endokaryotheca. This space is referred to as perinuclear space. Sometimes this perinuclear space might be filled with perinuclear fluid. Now, you can also find at certain places the exokaryotheca is continuous into an organelle that is referred to as endoplasmic reticulum. कई जगह पे exokaryotheca protrude out कर जाती है और एक organelle बना लेती है, that is referred to as endoplasmic reticulum, over which ribosomes might be present, and when ribosomes are present, then this organelle is referred to as rough endoplasmic reticulum. वो endoplasmic reticulum जो कि न्यूक्लियस के प्रोट्रूजन से बनी है और जिसके ऊपर राइबोसोम्स हैं, उसे हम बोलते हैं rough endoplasmic reticulum. Now, within the nucleus, there are more than 1,000 to 10,000 number of apertures which are present, commonly called as nuclear pore complex, and they contains certain proteins which are referred to as nucleoproteins. Each nuclear pore is made up of more than 50 proteins called as nucleoporins. Now, the organization of the nucleus, किस प्रकार से DNA और chromatin उसके अंदर organize है. So first was the nuclear envelope. The second is referred to as nucleoplasm. All the matrix which is present within the nucleus is referred to as nucleoplasm. Nucleoplasm is the one which transfers all the preformed mRNA into the cytoplasm. It appears that all the DNA which is being organized in the form of chromatin pours their mRNA into the nucleoplasm, and nucleoplasm flows out from the nucleus into the cytoplasm. ऐसा प्रतीत होता है कि सारे के सारे क्रोमेटिन मटेरियल्स अपने एमआरएनए को न्यूक्लियोप्लाज्म में डाल देते हैं और ये न्यूक्लियोप्लाज्म फ्लो कर जाता है न्यूक्लियस से साइटोप्लाज्म की तरफ, सो दैट द एमआरएनए कैन मूव आउट ऑफ द न्यूक्लियोप्लाज्म इनटू द साइटोप्लाज्म. द थर्ड most important structure is the nucleolus. Nucleolus is the darkly stained region which is present within the nucleus. You can find there are large, highly condensed or high density of chromatin material present here. This high density of chromatin material is because it is involved in the formation of rRNA. You know this rRNA will get associated with ribosomes. That means rRNA will get associated with proteins and will lead to the formation of ribosomal structure, and those ribosomes will be the one that will be produced here over the endoplasmic reticulum, converting into rough endoplasmic reticulum, ultimately leading to protein biosynthesis. So ribosomes are rRNA and proteins, and they are the one which are going to form the other protein molecules, protein biosynthesis. Now you can look forward that after the nucleolus, the next important structure is how this DNA is oriented. That is the chromatin. The DNA gets associated with certain proteins. When the DNA gets associated with protein, then it is referred to as DNA in association with proteins, especially the histone proteins and the non-histone proteins. So firstly, when it gets associated with histone proteins, then this structure is referred to as chromatin. Now, what I can tell you is that if the size of the nucleus is, if the size of the cell is about this tip of this marker, then the size of the nucleus will be approximately 1000 times less than the tip of this pen. Then what you can assume is that how can a DNA, the entire length of a chromosome, which is approximately 3 meters in length, can get oriented in such a small place? If I give you 46 threads and allow you to place them into a particular place which is as fine as the tip of this needle, अगर मैं आपको 46 थ्रेड दे देता हूँ 3-3 मीटर के और उन्हें बोलता हूँ कि इतनी सी टिप वाली जगह पे रख दीजिए, तो सारे थ्रेड इंटरमिंगल हो जाते हैं. तो क्या न्यूक्लियस के अंदर डीएनए भी इंटरमिंगल होता है? नहीं इंटरमिंगल होता, बिकॉज द क्रोमेटिन इज बीइंग ओरिएंटेड इन सच अ मैनर दैट इट फॉर्म्स अ कॉम्प्लेक्स विद द लेमिन प्रोटीन्स. लेमिन प्रोटीन जो एंडोकैरियोथिका पे है उसके साथ में क्रोमेटिन ऑर्गेनाइज कर जाती है और इस प्रकार से ऑर्गेनाइज कर जाती है दैट इट keeps the chromatin fixed. अगर हर एक डीएनए की पोजीशन, हर एक क्रोमेटिन की पोजीशन फिक्स हो गई, we can say that chromosome number 1 will be found only at a particular location within the nucleus. Chromosome number 2 will always be found at a particular location within the nucleus. Perhaps that is why when we shake our hand, the chromosomes are not getting intermingled with regard to each other because they are being separated from each other. जब हम अपना हाथ को हिला रहे हैं तो डीएनए क्यों नहीं मिल रहा? क्योंकि डीएनए को लेमिन ने होल्ड कर रखा है और ये डीएनए अलग अलग जगह पे हेल्ड है. लेमिन को होल्ड करती हैं दो और प्रोटीन जिसका नाम है लेमिन बी रिसेप्टर प्रोटीन्स एंड एमरिन प्रोटीन, और ये दोनों प्रोटीन ही एंडोकैरियोथिका के साथ में जुड़ी होती हैं. एंडोकैरियोथिका इज जॉइंड विद लेमिन बी रिसेप्टर प्रोटीन, एमरिन प्रोटीन, और एमरिन एंड लेमिन बी रिसेप्टर प्रोटीन्स आर बीइंग जॉइंड विद लेमिन्स. लेमिन in turns are being joined with the chromatin structure. The region of the DNA which is being joined with the lamin proteins are the adenine thymine rich region, and this is the heterochromatin region. वो DNA का region जो lamin के साथ में interact करता है वो adenine thymine rich होता है, heterochromatin region होता है, and that is why these DNA remain separated from each other. So this is all about the organization of the nucleus, the organization of our genetic material, chromatin within the nucleus. Now, please look here. There is a disease which is referred to as Hutchinson-Gilford progeria. अगर आप लोगों को ये पिक्चर याद हो, a character was there. That name was Oro. Now, Oro was suffering from Hutchinson-Gilford progeria. The famous legendary actor, the Big B, performed this role. The Hutchinson-Gilford progeria is a disease in which the aging is enhanced. In this disease, LMNA gene has a 150 base pair deletion. If this Lamin protein is not there, the organization of the nucleus is going to be gone. All the chromosomes are going to be intermingled. All the chromatids are going to be intermingled. अगर Hutchinson-Gilford progeria के बारे में आप लोगों को जान कराऊं तो एक पिक्चर थी पास जिसमें ओरो का रोल अमिताभ बच्चन ने किया था और उस पिक्चर के अंदर Hutchinson-Gilford प्रोजेरिया के बारे में बताया गया था। ये एक ऐसी डिसीज होती है जिसमें एजिंग बहुत एनहांस हो जाती है, पर्सन बहुत ही जल्दी एजेड दिखने लग जाता है। अगर हम इस डिसीज के बारे में स्टडी करते हैं तो इसके अंदर लेमिन ए जीन में 150 बेस पेयर का डिलीशन होता है, जिसकी वजह से लेमिन्स का डिग्रेडेशन होना शुरू हो जाता है। एक बार ये लेमिन डिग्रेड हो गई, ये नहीं बन पाई, तो अब क्रोमोसोम्स या क्रोमेटिन अपनी जगह पे नहीं रह पाएगा और आपस में इंटरमिंगल हो जाएगा। वही केस जो पेन के टिप के बहुत छोटी सी जगह में आपको बहुत सारे क्रोमोसोम्स को रखना था। नाउ द क्रोमोसोम्स विल गेट इंटरटेंगल्ड एंड they will lose their activity to perform protein biosynthesis. That is why new proteins cannot be formed at the place of older proteins and the person will suffer from progeria. So thanks a lot. This is all about the organization of the nucleus and the chromatin which is present within the nucleus. Now in our next lecture we are going to talk about the nucleolus. What is this nucleolus and what is the organization of this nucleolus within the DNA, within the nucleus and its functions. Thank you.